Hello guys, so for this week's Journal Club, I would like to go through this classic paper which describes the importance of the CD8 T cells in fighting against the hepatitis C virus infection. It is one of those, classic papers that, people who are, working in this field should read and refer to. So I hope you guys enjoy it, and if you have not already done so, please like this video and subscribe to my channel to help it grow. And without further ado, let's dive in. So this is the title of the paper, and please appreciate that the paper is old. It was written in 2003, but is one of the most cited papers in the field of Hep C. So let's dive in. So the aim of the study was to look at the role of CD8 T cells in Hep C reinfection. So there were two chimpanzees in this study. They were vaccinated with E1, the recombinant E1E2 glycoproteins. They were then challenged with the hepatitis C virus, and the CD8 T cells were then measured during the primary infection as well as during the reinfection 7 years after the first infection. So the role of memory T cells were also measured. So in this first result section, the authors wanted to look at the virus replication and immune responses. So they had two chimpanzees in this study, the CB056 and CB0572. So these chimpanzees were vaccinated with the E1E2 glycoprotein. So these are recombinant proteins, and the chimpanzees were then challenged with or infected with hepatitis C virus, and then the authors then looked at the replication of the Hep C as well as the immune responses. So if you looked at the gray shaded area here and here and here, you could see that at the earlier time points, so between day 0 to 50, you could see a rapid increase in the viral loads shown by the HCV RNA, and and what's interesting is that even though these chimpanzees produce the antibodies against the E1E2 at the earlier time points, so here and here, they still could not clear the virus, and it's only after they elicit like a T cell response, which is measured by ELISpot, which is a technique that measures the cytokine production, and in this case, they measured interferon gamma producing T cells. You can see that when T cells were elicited, as shown by the lines with the closed triangles, you could see that the viral loads were starting to drop in both animals. So this shows the importance of T cells. And I mean, what's also interesting is the serum ALT is the I think the serum ALT is highly correlated with the viral loads. So as the viral loads increase, I think the serum ALT, which is alanine aminotransferase, which is an enzyme in the liver, and and upon the liver damage, the serum ALT increases, and here it is the case. And in table 1, which is pretty much similar to what you see here, I think from what I got from the paper is that I think the data is derived from this table. You could see that so for chimpanzee 1 and chimpanzee 2, you see like a gradual increase in the viral loads in both cases, and then you see when the memory response kicks in, you see like a decrease in the Hep C virus. And what's interesting is that the memory T cells were elicited at day 56, as you could see here in the blood, but then it kind of disappeared in this animal anyway, kind of reduced as well. So it disappeared in the blood, and the author said, is probably because of the localization of these T cells in the liver. But nevertheless, the levels of the T cells, so the number of T cells, um, rebounded to a high level after day 99. So at day 121 and 149 post-infection, you see an increase in the number of T cells specific to Hep C virus, and that coincides with the reduction in the disappearance of the hepatitis C virus. And interestingly, it is it is interesting at the time, but because this was published years ago, a long time ago. So even after 7 years, you still see the memory response in these chimpanzees. So it's just a great study, I think. It's a great longitudinal study, and so using tetramer, we look at figure 1C now. So using the E2 tetramer, which measures, so in this case, they look at the memory T cells that were specific to this particular antigen or epitope, the E2, and the memory cells appeared at day 86 pretty much. So it's very early. So all in all, what this is saying is that T cells are important in controlling the virus and clearing the virus, and the memory T cells exist years and years after the first infection. So in this second results section, the authors want to look at the secondary infection. So the chimpanzees so after 7 years, the chimpanzees were challenged again with the same strain of the hepatitis C virus, and the virus replication as well as the immune responses were measured again. And if we just focus on the viral loads for now, shown in these like a shaded gray area here and here, you can see that duration at which the chimpanzees were positive for the virus or viremic was a lot shorter than the first infection, which lasted around 100 days and 150 days for one of the animals. And the reduction, so whereas here these animals remain viremic for only for just under 14 days, and the reduction in the virus tighter, kind of like coincided with the increase in the number of T cells as well as a T cell response. So these are memory cells. So basically what you can see here is that so I think after 10 days, I would say about 14 days, you see a very strong very strong T cell response in both animals. And you can see that in C and D as well. So so in the pre-infection, you get a small number, a small percentage of Hep C specific cells, but after 14 days, these cells underwent expansion, and you could see the increase in the percentage of these cells. And in figure 3, the authors looked at the responses in the liver. So they took some liver specimens, and so in A, so they took some liver specimens, and they expanded the CD8 T cells with anti-CD3 and some cytokines. And look at pre-challenge, you don't see anything because there was no expansion after 14 days, and 21 days you see a lot of CD8 T cells. And then these cells were used to measure the killing activity. So and as you could see here, for both animals, as the viral loads decrease, you see an increase of the killing activities by these expanded CD8 T cells. So which shows that the memory cells existed after 7 years, and they still have the killing capacity. Yeah. So in this result section, the result section 3, the authors wanted to look at the distribution of the CD8 T cells in the body of these chimpanzees. And so this figure 4, they looked at the T cell. They compared the T cell responses in the blood and the liver. And so this is the number of days or weeks after the second infection. So this is 28 days or 4 weeks after infection, 8 weeks after infection, and 24 weeks after the infection. And they looked at the E2 specific, which is the glycoprotein specific CD8 T cells. And as you could see, that at all the time points, there were more of these E2 specific CD8 T cells in the liver than in the blood. So these are, they're tetramer. So cells that were specific to this tetramer here. They then looked at the expression of CD69, which is a marker, which is a marker for T cell activation. And as you can see that so 8 weeks and 24 weeks after the second infection, but if you were to because it took them about 2 weeks to clear the virus, these were 6 weeks post clearance, or 22 weeks post clearance. You could see that the CD8 T cells in the liver still express CD69, whereas in the blood, pretty much none of the T cells, maybe 1 or 2, express CD69. So what this is showing is that in the liver, there's a constitutive expression of CD69. so they remain activated in the liver. So in this figure, so the authors wanted to assess the importance of the CD8 T cells in controlling the virus. So what they did was they administered the anti-CD8 antibodies, which deplete CD8 T cells. It's an antibody that deplete CD8 T cells prior to infection. So if you just look here, so this is when they gave the virus to the chimpanzees. So at this date here, so on this date, there was no CD8 T cells in both animals, and then and throughout the course of infection, they measured the virus as well as the immune response. And as you could see in B and C, so if we just focus on the gray area here and here, you could see that the chimpanzees struggle to control the virus, without the CD8 T cells. And this kind of coincided with the lack of CD8 T cell response, so lack of interferon gamma producing T cells, but only when you get like a partial restoration of CD8 T cells on day 42, which can be shown here. So here and here, that's when you get the that's when you see the reduction and clearance of the virus. So this figure highlights the importance of CD8 T cells in controlling the virus infection, and it's basically, and also one of the things to keep in mind is that like these represent the response in the blood but not in the liver, so there might be more of CD8 T cells in the liver, but they just did not check because it is very hard to check the to measure responses in the liver. Yep. So this is the last figure of the paper, and for me, this is the is the figure that summarizes the whole study. So you have two chimpanzees, right? And after the primary infection, which is the gray, the gray curve here, you could see that the primary infection is characterized by like a prolonged viremia, or prolonged positivity for the virus before clearance, as shown here. And so after the clearance and after 6 years, the chimpanzees were then challenged again with the same virus. But this time, so if you just look at the like a darker gray curve, you could see that the animals remain viremic for a very short time, so for only 2 weeks. So they cleared the virus within 2 weeks. So that's the memory response. And then you have the third curve, which is as shown by this dashed line here. So if you were to deplete the CD8 T cells, it would take a lot longer for these chimpanzees to clear the virus, and so only when you get the restoration of CD8 T cells, that's when the chimpanzees were able to clear the virus. So basically, this study highlights the importance of CD8 T cells, and it also shows that the kinetics of the memory response to hepatitis C virus.